So we're going to start in about 10 minutes time, isn't it? Am I correct?
Yes, sir. Yes, sir. We're going to start in 10 minutes, sir. And today we're expecting some doctors also. And I invited one doctor. He is working in Apollo hospitals in Hyderabad. He is Dr. Victor, MCH. So I requested him to join the session because I have we have small cordial relationship. So I requested him, if you permitted after the talk, he would like to talk to you for a couple of minutes.
Right. Yep. Most happy to. Yeah. Yeah. I was in Hyderabad. I think probably 4 years ago.
Oh, great.
Yeah. Really nice place. I love the fantastic food.
Yes, sir. It's biryani famous here.
Yeah. Exactly.
Yes, sir.
I wish I can visit Hyderabad again, but not
definitely,
sure.
Sir, you're most welcome, sir, and give us chance to provide you a good hospitality in Hyderabad.
Sure. Yep. Most important is that we just keep in touch.
Definitely, sir. And the 3 prestigious colleges and the 100 years old colleges are organizing this International FDP Programme. And that is one Nizam College. It is a 132 years old college. And Government City College, 100 years old college. And Loyola Academy is more than 90 years old college. So these 3 are the autonomous colleges, and these 3 are the best colleges. So, and we are extended our support together, and we're organizing this International FDP Programme. And the State Council of Higher Education Department of State also supporting this program. And we collaborate with the Federation of Asian Biotech-associated. It's a non-profit organization. Every year, the FABA organizing the BioAsia in Hyderabad. So they're acting as a knowledge partner. Yes, sir. They're acting as a knowledge partner for this program.
Amazing. I mean, pulling so many partners and organizations together.
Yes, sir. It has happened because of the eminent speakers like you, because people see the profile of the speakers and they'll come forward and join together, sir. Thank you for accepting our invitation once again.
Thank you. Thank you very much.
And another institute, sir, BCIL, Biotech Consortium India Limited. They are also supporting this program and advertising this program in the global platform. And exclusively, Dr. Rao is supported very much for making this FD program in a smooth manner.
Yeah, he's a really a good man. We have known each other for a couple of years now. Haven't been seeing him for a while, other than online. But he's a good man, yes, and he still maintain a close tie with you all, isn't it?
Yes, sir.
And I think Dr. Rao also he frequently visits India, speaking for you all as well. Am I correct?
Yes, yes, he monthly once he'll going to get to talk to the Hyderabad institutions and Indian institutions, sir. Now the entire world is looking on the gut bacteria, sir, because recently, if you see all the research publications and research thing, everything. So while discussing with Dr. Rao, and he asked me what kind of areas you are looking for this continuous FDP program. So I said that, sir, the biotechnology and life science having the vast applications, and most of the faculty who are working in, especially in India, our education system is different when it comes to the foreign education system, because the Indian education system won't allow the foreign faculty to teach their students or to train their faculties, because we have certain policy restrictions. But when it comes to yesterday, we had a fruitful discussion with Dr. Rao, because the Malaysian government allows the public-private collaboration. So that is the main reason for, it's the main hurdle for it, because once the policies are, again, they change the policy decision, then only it will be useful for everyone that we can exchange our knowledge and research collaborations for the future. That's what we're expecting. So after, yes, sir. Yes, please go ahead, sir. Yeah.
yeah, I mean, I totally agree with you. There's always this disconnect between the government and the industry, but the bridge between the two is actually very important. In fact, it was a missed opportunity for us in Malaysia. The academia is working in silo, and the government is working in silo. The private is working in silo. So everyone is working in silo. There's no bridge connecting, each other. But it's not easy to build that bridge as well. It requires a lot of commitment, time, and, as you mentioned, policy.
Yes, sir. That is the first thing, sir. Because until unless you open the door, you can't go outside, or who can't will not allow others to inside. The policy decision is the first. They should change the policy decision, but it will be very tough because the government institutions are lack of funding and resources.
Yep, it's always it's always resources and always time. The same excuse.
Yes, sir. It is everywhere, I think so.
Yeah, it is. I mean, even in Malaysia itself, we are very far from being perfect. And likewise in the developed countries, where they have more resources, it's also quite difficult to achieve that kind of collaboration. But obviously they are more matured than us.
It's true, sir. So while discussing the trial, so I asked him, sir, I need the latest areas that where the science to be explored. And after I suggested him, sir, I would like to have a talk on the microbiome sciences. Then he advised, I have a good friend in Malaysia, he is an eminent speaker, he suits for your talk. So I said that it will be very good. It will be grateful to you if you provide the information of the professor that we are saying. So he advised us your name, and we accepted the invitation, and we are very thankful for that one, sir.
Yeah, thank you once again. But I'm sure you're aware that I'm actually a clinician, right? And
yes, sir, yes, sir.
Yeah, I do see patients at the hospitals, but I'm also a researcher and a scientist as well, and certainly working on this area. With some, I do see challenges. Now the thing is like, I think the talk that I'm going to give is more of mentioning what would be the progress, the updates, and knowledge about the microbiota sciences.
Yes,
but hopefully during the discussion we can discuss a little bit more detail on how biotechnology and collaboration between academia and industry can come in.
Yes, sir. Yes, sir.
All right, so we still have 2 more minutes to go.
Yes, sir. Yes, sir. Yeah. Sirisha, madam, hope you are there. Yeah. Now we are going to start in 1 minute, so make things ready. And
good afternoon,
Professor.
Namaste.
Perfection is not attainable, but if we chase perfection, we can catch excellence. A very good afternoon. to one and all present in the meeting. Esteemed fathers, speakers, and our lovely audience. A very warm welcome to the 4th day of the FDP program, which is themed Microbiome Sciences. So without waiting any longer, I would request Dr. C.H. Sirisha, Dean of Academics, PG Loyola Academy, to deliver us the welcome speech.
Education is not the learning of facts, but the training of the mind to think. Rightly said by Albert Einstein. Good afternoon to one and all. We all know the importance of science and technology innovations in our day-to-day lives and the ways in which they are transforming the world. I'm confident that you will find new ideas and novel partnerships to sustain your efforts in support of transforming life science research. With this contest, Nizam College, in association with the Government City College and Loyola Academy, came forward to organize virtual faculty development program on recent trends in transforming life sciences, focus on academia and industry symbiosis. At this juncture, I, Dr. Sirisha, Dean of Academics in PG, Loyola Academy, takes this precious opportunity to welcome all the guests for today's event. It's a moment of pleasure to welcome all distinguished speakers, both in India and abroad, esteemed guests, faculty members, and research scholars to this 4th day of International FDP. Today's theme is Microbiome Sciences. I extend a hearty welcome to today's speaker, Professor Yeong Lee, President-elect, Malaysian Society of Gastroenterology and Hepatology. We welcome you, sir. I would like to extend a warm welcome to FDP co-convener, Reverend Father C.H. Anand Kumar, S.J., Vice Principal, Loyola Academy. We welcome you, Father.
Loyola Academy is known for academic excellence, innovation, and research. To know more about this prominent institution, I request Reverend Father C.H. Anand Kumar, S.J., Vice Principal, Loyola Academy, Hyderabad, to give us a brief information about Loyola Academy.
Thank you, Madam. Loyola Academy is run by Jesuit fathers. Jesuit fathers who also run many other reputed schools, colleges, and universities in India and around the globe. All these institutions share the vision of imparting education with integral formation that involve academic excellence, spiritual growth, social commitment, and value-based leadership. And the mission of forming men and women for others and molding our students as global citizens and with competence, conscience, and compassionate commitment. Loyola Academy was founded in the year 1976. Initially, junior college was established and later UG and PG colleges took shape. The college is blessed with spacious campus of 132 acres that has, apart from huge academic buildings, hostels for boys and girls, several play fields, and a beautiful research farm. The college was granted autonomy in the year 1992. It was awarded the status of college with potential for excellence in the year 2008. The college was accredited and awarded A grade by NAC in 2005 and 2011. It was re-accredited with A grade for third cycle recently in the academic year 2019-20 and granted an extension of autonomy for 5 years. One significant fact that is worth mentioning about the college is that it has been offering from its very inception only professional and job-oriented courses. Currently, the college is offering
28 UG courses and 7 PG courses. More than 5,000 students are studying in the campus. There are about 170 teaching staff and 60 non-teaching staff working in the college. During the pandemic, online classes have been conducted successfully using Microsoft Teams original version that is purchased by the college. Semester and online examinations also were conducted successfully online. About 40 choice-based certificate courses and 2 non-CGPA compulsory certificate courses are offered by the college. College doesn't miss expressing its solidarity with the poor and needy. During the second wave of the corona pandemic, more than 500 families in the slums close to the college were distributed free and quality food packets every day for 2 weeks. Snacks were distributed to police in 95 checkposts across the city for 10 days during the lockdown. And past several days, a huge vaccination drive is going on in the campus. We are making sure that all our students, teaching and non-teaching, and contingent staff are vaccinated. A brief mention of our biotechnology department. It was established in 2003. The department boasts of excellent labs that are equipped with sophisticated infrastructure and the state-of-the-art facilities, with MOUs with international research institutes like IIMR and several biotech companies. The department boasts of more than 80% placements with excellent packages every year. With this brief introduction about Loyola Academy, I wish and hope and pray that all the organizers and participants of this International Faculty Development Program will immensely benefit and objectives of this program will be realized. I thank our Principal, Reverend Father Dr. L. Joji Reddy, Correspondent, Reverend Father Dr. S. Emmanuel, and our Dean of Sciences, Dr. C.H. Sirisha, for giving me this opportunity to speak about our college. Thank you very much.
Yes, indeed, Loyola Academy is true to the words spoken by our noble Vice Principal. We are honored and privileged to have you as co-convener of this International FDP program. Thank you, Father.
Thank you, Father.
We now have Mrs. D. Gurudevi, who will be introducing us to our esteemed speaker for the day.
Good afternoon, one and all. Myself, I am Guru Devi, faculty in Department of Biotechnology, PG, Loyola Academy. Firstly, I would like to thank the organizers for giving this opportunity to introduce our guest speaker to august audience. Moving right along, it's now my honor and privilege to introduce today's distinguished speaker and eminent scientist, Professor Yeong. He is a professor of medicine and consultant of gastroenterology, hepatology, and internal medicine of University Sains, Malaysia. After his gastroenterology fellowship, he completed his PhD in Glasgow, United Kingdom, and later a postdoctoral fellowship in the United States. Professor Lee is currently the editor of BMC Gastroenterology, PRJ, Journal of the Royal College of Physicians of Edinburgh, and the Malaysian Journal of Medical Sciences. Added to his credentials, Professor Lee has published several papers in high-impact journals, including Gastroenterology and Gut, in several book chapters, and numerous articles in conference proceedings. As a feather in crown, JCI Malaysia awarded him the Outstanding Young Malaysian Award in 2015 and the Top Research Scientist of Malaysia by the Academy of Sciences, Malaysia in 2018. His current research interests include gut microbiota, neurogastroenterology, functional gastrointestinal disorders and cancer, gastroesophageal reflux diseases, obesity, and endoscopy. Presently, Professor Lee is the President-elect of the Malaysian Society of Gastroenterology and Hepatology and a committee member of numerous international societies like World Gastroenterology Organization, OESO, and the American Society of Gastrointestinal Endoscopy, and also the Scientific Chair of Asia-Pacific Digestive Week 2020. Indeed, we are delighted to have you for today's technical session, and it's our pleasure to welcome you, sir. Now, without any further ado, we will turn the time over to Professor Lee. Thank you one and all. Thank you, madam.
Thank you very much. And it's really a fantastic day for some of the very hopefully interesting discussions to come. I would like to share my slides.
Sir, you are in mute, sir. Unmute yourself, sir. Yeah, thank you.
I actually already unmute, so I hope you all can hear me clearly.
Yes, sir.
All right, so let me just share my PowerPoint. I hope you all can see my PowerPoint.
Yes, sir.
Okay, great. So again, I'd like to extend my sincere thanks to the organizers, also Dr. Simba, whom I've been talking for a while before the start of the session. And I got to know many great things about the International Faculty Development Program and how difficult is it, to organize a meeting involving so many partners. I hope I can, bring on the title of the microbiome sciences to my title today, the gut microbiota and little brain, big brain. Now, little brain here means the gut, and the big brain here means the obviously our central nervous system. And the reason for gut being a little brain is that it is really the second largest neural network after the big brain. And all of these are intricately connected. So before that, let me just outline my presentation. I will, first of all, touch on the gut microbiota. Secondly, it will be on the gut-brain axis. And as a clinician, I would certainly like to, talk upon the disorders that are associated with a disrupted microbiota gut-brain axis, and what could be the potential therapeutic application, which hopefully will provide some input for the academia and the biotechnology, which are the main stakeholders today. As a clinician, I would always like to start with a case. This is a patient that I have seen in my practice, a 52-year-old female with a body mass index, the BMI of 39. So she would be categorized as obese. And she has a diabetes mellitus as well as hypertension. Since young, she has been complaining of intermittent abdominal discomfort as well as bloating. So bloating would be a sensation where you feel like gas in the stomach. And her doctor diagnosed her with a Helicobacter pylori gastritis. Now, Helicobacter pylori is a bacteria found in the stomach, and it really has been around with us for thousands of years since the existence of human beings. And gastritis means that she has inflammation in her stomach. And then at the age of 48, she developed progressive hand tremors and rigidity. And upon investigation, she was diagnosed with a Parkinson's disease. Now, Parkinson's disease is a neurological disease, which, as mentioned here, characterized by hand tremors, and is due to a dysfunction in the brain, causing many of these motor disorders, not just the hand tremors, but may also sometimes involve difficulty in the walking and so on. Now, last year in April 2020, her GI symptoms, GI stands for gastrointestinal, worsened with diarrhea as well as a nausea. And she turned out to be a COVID-19 positive, a pandemic or the disease that really affect all of us. Her GI symptoms recovered but persisted, and her Parkinson symptoms seem to fluctuate more despite being on a levodopa. So a levodopa is a medication that really helps or to treat her Parkinson. Now, maybe you might ask, what really is the role of gut microbiota in all this? Now, we are in a really rapidly changing landscape. During the early days, poor hygiene is often a reason for many of the infectious diseases that we are seeing, including Helicobacter pylori. In fact, Helicobacter pylori itself is the main cause for peptic ulcer disease, as well as gastric cancer. And because of economic development, there has been a rapid change in the diet. And of course, this also leads to increasing trend in obesity. And this leads to many chronic metabolic disorders that we see today, including diabetes, as well as cardiovascular disease. Now, the gut microbiota here is of utmost importance. We got to know about this more recently. And the balance of this gut microbiome, the so-called, to the lay people, will be the bad bacteria and the good bacteria. The balance between the two is really important for gut health. I did mention that Helicobacter pylori itself is an ancient microbiome that exists together with human development. And this is really something that is a paradox, because we always think that the human stomach that contains acid can practically kill any bacteria that exists in the food and so on. So little do we know that peptic ulcer disease was actually due to an infection. Before we know the existence of Helicobacter pylori, we always thought that peptic ulcer disease is due to stress. But thanks to Warren and Marshall, they found this pathogen in the stomach back in 1982, and they won the Nobel Prize in 2005. For those who do not know Warren and Marshall, Marshall himself, he found a bacteria during his student time, being a medical student in the medical college. So they subsequently also proved that the bacteria doesn't just exist in the stomach, but it's also a proven cause for peptic ulcer disease and stomach cancer. If you know, peptic ulcer disease and stomach cancer is really a major public health problem in the Asia Pacific, where Helicobacter pylori tends to be found in almost half of the population in China. But interestingly, in India, we call it as an India paradox, where although the Helicobacter pylori infection is high in prevalence, but the stomach cancer is not as common. So this is somewhat of a paradox and currently still in investigation. Now, Helicobacter pylori itself is found to be prevalent among Malaysians with Parkinson's disease. In fact, about 1.5 to 3 times more infection in Parkinson itself. And those Parkinson patients who has a Helicobacter pylori infection also display worse motor functions. And by eradicating the infection, it also helps improve the motor function and also improve the levodopa absorption, making it more, better in terms of treatment. Now, the mechanism of how a Helicobacter pylori and Parkinson's disease remains unclear, but there may be some direct neurogenic inflammatory process, or there may be certain functional changes along the GI tract and also the gut-brain axis, as shown in the figure here, which I will explain a little bit more in my subsequent slides. So let's talk about gut microbiota. Now, there has been a lot of interest on the gut microbiome. In fact, we know that there are pathogens in the GI tract, but in the older days, the only means for us to detect the pathogens are from what we call as culture. Now, culture is a tedious and long process. In order to grow the bacteria, it may take several days, but the culturing process in the lab, has advanced in such a way that we can identify the bacteria within hours. However, we still do not exactly know what could be the profile of other bacteria exist in the GI tract. And this is where the modern molecular technology make it possible, where we can use a sequencing technology in order for us to sequence the DNA exist in the GI tract of those bacteria. In fact, if you look at the diagram over here on the right side, there are all sorts of bacteria that exist in the stomach, in the small bowel, and as well in the large bowel. The numbers of bacteria that exist in the stomach will be less because of acids, obviously. And the number grew along the tract. Again, less in the duodenum or the proximal part of the small bowel. But as you go along the distal part of the tract, there are more and more bacteria. And the most bacteria exist in the large bowel. Now, the balance in the composition and the abundance, these are the two terms that we tend to use to denote what are the types of bacteria exist, i.e. composition, and how much of it that exists, and this is how we term abundance. So a balance in the composition and abundance will lead to what we call as a eubiosis. Compared to an imbalance, then this is what we term as a dysbiosis. Now, it's very interesting to know that gut microbiota has greatly evolved, right? Where there is a changes at different stages of life. Many may think that the baby is supposed to be sterile. The gut in baby is supposed to have no bacteria, but more recent study has found that this is actually not true. And within the first 1,000 days of the infancy period, this is the time when the gut microbiome really plays its most important function in the development. In fact, if you were to really modulate this time period, the first 1,000 days, it will lead to a much healthier adult in the later part of the human development. And most of the time, by the time a child reached the age of 3 years old, the microbiome diversity of the population tends to be more stable. And in adults, it is also stable unless there's a change in diet or environment, or with the use of certain drugs, for example, antibiotics. And by the time we reach our elderly, then the diversity will be lower. And all of this may actually cause, short or even long-term health effects. So again, what are the determinants, or what are the factors that influence the health and diseases through the gut microbiota in early life? So a group of us, we decided to, come up with a position paper, such as this, published in the Journal of Pediatry and also Child Health. We screened through the literature, we talked to experts in the field, and these are some of the statements that we came out. One is that the colonization of the gut microbiome actually begins in early life, as early as in the womb of the mother. The cesarean section itself can be associated with the altered gut microbiome colonization. Compared to a vaginal delivery, if someone who delivered by cesarean section, the chances for an altered gut microbiome is much higher. And there has been some studies showing that with cesarean section, it tends to be associated with a more obese child. And breastfeeding and infant formula have very different impact on the gut microbiome. Obviously, breastfeeding would be much more better, more natural microbiome exists compared to infant formula. Nowadays, many of the infant formula have also incorporated probiotics, trying to simulate breast milk. But to this date, I think the biotechnology companies are still, trying to find the most perfect infant formula that really simulate breast milk. So the use of antibiotic during pregnancy and infancy can affect gut microbial colonization. And more importantly is the environmental exposure. We call it the exposome. Many things in the environment can change or modulate the gut microbiome, including when you watch your most favorite movie star on the TV, it can excite your gut microbiome. You watch a football, you can also change your microbiome transiently. So what could be the impact of a dysbiosis in disease-wise? So there could be a short-term impact or the long-term impact. So a short-term impact for some patient, they may have diarrhea or even what we term as a Clostridial difficile disease. In the longer term, it may lead to obesity, metabolic disorders like diabetes, even gut diseases like inflammatory bowel disease, allergy, or even certain psychological disorder like depression and autism. So you may ask that what really determines the composition of the gut microbiome? Is it the diet or the genes? So this was a study that we performed in collaboration with the National University of Singapore, the top university in the Southeast Asia and also part of Asia. And what we did was to administer a food frequency questionnaire and collect stool from the primary school children, as you can see the pictures over here, where we subject the stool to metagenomic analysis. We recruited primary school children aged between 7 to 11 years old, with almost quite a balance male and female children of two ethnicities. Obviously, they would have different genes, and we found that about a quarter of them being obese. We also compared the microbiome changes with those of the Chinese in Guangzhou. And what we published this paper in a scientific report. You're more than welcome to read this paper. But what interesting was that we found the Malays and the Chinese, despite having different ethnicity, if they are in the same region, they would have a similar microbiome profile. But Malays and the Chinese, who are from a different part of Malaysia, they also share similar microbiome despite they being different ethnics or different genes. So obviously they share the same diet, and that's the reason why they those Malays and Chinese would have similar microbiome profile. And therefore enterotype of a population is largely determined by diet and not ethnicity. You are certainly what you eat. You may also ask which particular diet is beneficial. So we published this particular paper in JACI. JACI is the top journal in allergy and immunology, where we published this review paper, and there's been a lot of hype with regards to Mediterranean diet. In fact, Mediterranean diet is very similar to our own traditional Asian diet, which is very rich in fiber as well as fermented or functional food. For example, in India, it would be yogurt and so on. Now, with high fiber, which contains prebiotics, and functional food which contains probiotics, they would promote more beneficial microbiome in the gut, especially the Prevotella, which is fiber degrading microbiome, and it can produce more beneficial metabolic byproducts, the butyrate, which has been shown to be anti-inflammatory. But the problem is that that we face nowadays is the use of drugs, including antibiotics. In fact, antibiotic prescription is on the rise, causing a lot of anxiety in terms of antibiotic resistance and so on. Like based in this study in Malaysia, where when they surveyed the clinics, more than 21% of clinics actually prescribe antibiotics at any time, more in the private practice, obviously, and almost half of the indication are for upper respiratory tract infection. There's a lot of uses of penicillin and cephalosporin, accounting for more than half of the prescriptions. Now, little did doctors know, antibiotic-associated diarrhea is really quite common, very commonly reported by patients, but seldom seek any consultation. And very often in antibiotic-associated diarrhea, it tends to occur about 2 months after end of antibiotic. But one of the main reasons why there's a diarrhea after antibiotic is because of the dysbiosis, because it disrupts the gut microbiota balance. So let's go back to the patient, a case study where this is a patient with a 38-year-old male, no previous medical illness, who have dyspepsia, which is always abdominal discomfort. And he underwent an upper endoscopy and diagnosed with a Helicobacter pylori gastritis. And he was started on antibiotics, including amoxicillin, clarithromycin, and a proton pump inhibitor for 2 weeks. So the question is that, does antibiotic have any effects on microbe in the stomach? So at this juncture, I assume that everyone would say yes. And in fact, the study have shown that by giving antibiotics, it does cause changes in the abundance, right, of the microbiome within the next 7 days. Now, the question is like, does this balance will recover after stopping the antibiotics? It does. Some of them actually did recover. If you have seen, the chart here, for example, the firmicutes, despite reduction during the first week after antibiotic, it does recover, and in fact, back to the baseline. But there are some beneficial bacteria, like the actinobacteria, after the antibiotics, although it's trying to recover, but it never recovered back to the baseline. And this can last for several months. Studies have also shown that by giving antibiotics, it does cause a reduction in the alpha diversity. And this term is a commonly used term where what it means is that there is a reduction in the composition in terms of number of species. Now, say for example, before the antibiotics, you can have 100 species, but after the antibiotic, it reduced by 50 species. So this is what you mean by a reduction in the alpha diversity. Now, also in this particular study, which is quite interesting in the normal population, where whether there's any difference in giving antibiotics, or you give a probiotics, or you give antibiotics followed by a probiotics. So basically what it shows is that if you were to give antibiotic, certainly it will change the microbiome, and it's really difficult to recover, right? But if you were to give a probiotic first, then the changes or the effect of antibiotic will be lessened with the use of probiotics. So just a little advertisement. If you're interested in neurogastroenterology and motility, this will be the textbook that I have written together with my colleagues, Professor Satish Rao, as well as Professor Uday Ghoshal. So let's talk about the gut-brain axis, the little brain and big brain. Now, the gut-brain axis really is a very complex bidirectional communication between the central nervous system and the enteric nervous system. So in the composition of this axis include not just the central nervous system, but also a neuroendocrine, neuroimmune cells, autonomic nerve, and as well as a microbiome. So the the gut-brain axis itself has common development because they do have a common embryonic origin. If you look at this particular diagram, by week 12, the neural crest cells actually migrate into the wall to form the enteric nervous system. So they are sharing same neurotransmitters and receptors. However, the effect of this neurotransmitter or receptor may differ slightly between the two. For example, if you were to give a drug that affects the 5-HT, which is the serotonin, in the central nervous system, it may treat depression, but the serotonin effect in the gut will cause diarrhea. So it's very important to understand that certain therapy or drugs that act on certain receptors could have differential effect on the central nervous system. Again, if you were to inhibit a 5-HT reuptake, meaning there are more 5-HT, it would have an antidepressant effect in the central nervous system, but diarrhea in the gut. If you were to inhibit the norepinephrine reuptake, then it will cause, it has an antidepressant effect, but causing constipation in the gut. If you were to inhibit a dopamine reuptake, it will cause an active central nervous system effect and nausea in the gut. 5-HT1 agonist, it has antidepressant effect, but it may cause nausea, headache, nervousness, and so on with a 5-HT3. So what are the components of the enteric nervous system? Let's talk about the sensory first. There are a lot of sensory afferents or receptors within the gut itself, as you can see here. There are also some sleeping pain receptors that can wake up in response to injury. And most important receptors are those found near to the lumen, where they actually sense the chemical that are absorbed across the mucosa. Now, why is knowledge this is important is because there are two very common symptoms that are that may result from disturbance in these receptors. One is which is allodynia, where the stimuli that normally are physiological causes pain, or hyperalgesia, where the responses that are painful become exaggerated. Now, this concept of allodynia and hyperalgesia leads to two further concepts, what we call as the peripheral sensitization and the central sensitization. Now, for peripheral sensitization, basically the damaged tissue from the inflammation will activate all of these receptors, and this will result in what we call as a visceral hypersensitivity. Now, I must say that many of our patients say, I'm sure many of us would have experienced abdominal discomfort, say after meal, but some certain patients will experience more discomfort. And the reason is because their gut is more sensitive than other people. How do we characterize sensitivity is that, for example, in a normal person, after meal, they produce a certain number of gas. But for those who have a sensitive gut, even with a small amount of gas, they can feel even more pain. So this is what we mean by hypersensitivity. Now, for central sensitization, it's the same concept as the so-called peripheral sensitization, but this central sensitization takes effect in the central nervous system. And the main reason for having a central sensitization is because there is what we term as a neuroplasticity, right? So the brain itself is a very unique piece of tool or a piece of machine that we have that really can have a lot of plasticity. It's very interesting. Now, how does the enteric nervous system organize? It's really interesting to know that in the gut itself, everything is like have been well programmed. It's like a mobile applications. So, for example, you know, in the small intestine, you have this so-called postprandial app. You can also have an interdigestive app. You can have a boral power propulsion app, and you have an emetic app. And why is this app important? Is because, say for example, if you have taken a wrong food and your gut sense that a chemical is not suitable, it needs to activate the app in order to propulse it outside, you know, to reject the food itself. And it can be shut on and off through the working of interneurons. There are also very interesting cells that we need to know. For example, the interstitial cells of Cajal. This is really the pacemaker cell for the small bowel, and it really governs the contractility of the small bowel itself. We also need to know that within the mucosa itself, there are chemicals that allows a very integrative working, you know, between the mucosa layer and also the deeper muscle layer in the gut. For example, in the mucosa layer, there are what we term as enteroendocrine cell, or the EC cells, that produce serotonin. And this serotonin is the most important neurotransmitters within the gut. And many of the drugs that act on this will really have an effect on the motility. So likewise, when this serotonin acts with or interact with the nervous system within the deeper layer of the gut, it would have either a secretory effect or a motility effect. So secretory effect is obviously important to for the gut to produce the secretion, as whereas for the motility effect, it's important for the movement, or what we term as the peristalsis along the gut tissues. Other than that, in the gut, there's also a lot of immune cells, and any of the inflammation can actually, cause immune activation in the gut because of the close connection between the immune cells and also the nerve cells in the gut itself. Now, I think a lot of people may not know that there are actually receptors that exist on both the immune cell and nerve cell in the gut. This is what makes the gut actually quite unique. And they have differential effect either by inhibiting or by activating it. For example, if you have to activate the vagal cells, it can have anti-inflammatory effects through the mechanism by the macrophages, and also the cholinergic effect and so on. Now, the most important probably is the window between the gut as well as the body, which is obviously the intestinal barrier or intestinal mucosa. So this is the intestinal mucosa is really the window or the interface between the external environment, which is here, and the internal environment, which is the body, right? So the gut microbiota itself and the food antigen, these are the two main sources of interaction between at the interface between the external environment and the internal environment. Like and not just the interaction, but many of this interaction is by activation or modulation of the immune cells, as well as the crosstalk with the enteric nervous system. So hopefully there's not too much of a knowledge there. It's quite technical, but if you have any question, you can always ask me during the Q&A. Next, we'll talk about the disorder that would disrupt the so-called microbiota gut-brain axis. So this brings me to another case. Sorry, the same case that I mentioned early on, right? And so she actually developed a COVID-19, and the GI symptoms although recovered, but her Parkinson motor symptoms seem to fluctuate. So you may ask, what could be the connection between the Parkinson disease, the COVID-19, and the gut microbiome? I think many of us by now would know that how the COVID-19 causes some of these GI problems in the gut. And lastly, it's because the virus itself acts on the ACE2 receptors. And very interestingly is that the ACE2 receptors are found in abundance in the GI tract. And therefore, many of the patients with COVID-19, they do have gastrointestinal symptoms. And if they do have a gastrointestinal symptoms in COVID-19, it may be a sign that the COVID-19 disease itself could be more severe. So how does the gut microbiome comes in? In fact, with the virus, the COVID-19 virus, it somehow can cause a dysbiosis in the GI tract itself, and this interact with the enteric nervous system, and therefore also cause some brain effects, either through a direct invasion or through a blood-brain barrier breakdown, or there could be some immunological effect that we are not sure. So we do see that COVID-19 can cause a dysfunction in the gut-brain axis. So how does dysbiosis result in some of these degenerative disorders or neuropsychiatric disorder that we see? So some of these disorders include depression, schizophrenia, bipolar disorder, Parkinson, autism spectrum disorder, the attention deficit hyperactivity disorder, Alzheimer's, multiple sclerosis, anorexia nervosa. All of this have been found to be associated with changes in the gut microbiome itself. So it means that some of this microbiome may be a potential biomarkers for the biotech company to explore. Although I would say that how these changes leads to the disease are still unclear. So therefore, we do need more mechanistic studies in that regard. And the next question is like, how can we intervene through the modulation of this microbiome? Obviously, this can probably be done by the use of probiotics, or even what we term as a fecal microbial transplantation, which I will mention a bit more after this. So let's talk about the Parkinson's disease itself, and how does this dysbiosis can lead to Parkinson. Really, there's been quite a number of recent studies that have really put into this perspective on how dysbiosis, including including a lower prevotella and also lower production of butyrate, can lead to increase in the amyloid-like fiber producing microbes. So when there is a dysbiosis, basically it leads to increase in the amyloid fiber producing microbes. So these are the microbes that will lead to production of amyloid fiber that then travel along the vagus nerve here, and gets deposited in the brain, causing the Parkinson disease. In fact, this is what we call as alpha-synuclein. These are the ones that really migrate to the brain using certain routes that I've shown in this diagram. And it has an ability to even sit like a prion-like. So if you know, prion is another type of virus where it can spread in the brain, causing neurodegeneration. So the vagus nerve has been found to be a main pathological route. It has been found that also by doing a truncal bichotomy, meaning that if you cut off the vagus nerve, it actually reduces the risk of Parkinson's disease. What about Alzheimer's? In fact, in Alzheimer's, the dysbiosis feature is quite similar. It has a low butyrate producing organism and a higher a myeloid producing organism. And because of that, it leads to a loss of these cholinergic neurons in the enteric nervous systems. And all this tau and amyloid protein, these are the two proteins that get deposited in the Alzheimer's disease, get phosphorylated or aggregated, and this also causes a prion-like seeding and so on. But unlike a Parkinson, if you were to do a truncal vacuotomy, it doesn't seem to reduce the risk of dementia, which is quite interesting. And again, further investigation is needed. So what about COVID-19? We know that obviously respiratory manifestations are also common, is the most common, but we also understand that the GI and the neurological symptoms are equally important. In fact, they are quite common with incidence between 50 to 79%. Some of these symptoms or GI symptoms include anorexia, which is exists in 46.5%, but there's also nausea, vomiting, diarrhea, as well as abdominal pain. As for neurological COVID, there are basically three forms. For example, there's a psychological form, there's also a chronic fatigue symptom form, and also more of a catastrophic form, which may include stroke or encephalitis and so on. So all of this will lead to long COVID-19. Basically, long COVID means that after the initial COVID-19 infection, these are some of the potential long-term effect after the resolution of the COVID-19 symptoms. As I mentioned as well, that some of these GI and neurological manifestation may actually means it could be a more severe, it may mean a more severe form of COVID-19 infection. So in this particular study, it has shown that those who have developed COVID-19 and also Parkinson's disease, they have actually worsened motor function and also require more levodopa. So this is basically what it means. And it does help to explain why that my patient who has a Parkinson and developed COVID have more severe symptoms. Okay, now let's move to another patient, a 49-year-old female, single with a BMI of 39, with allergy, hypertension, and fatty liver, no family history of colon cancer. So she tried to lead a healthy lifestyle, but no time because of work. You know, nowadays in our society, many of our younger people, they have just no time. They just have to work, and they have very difficulty in leading a so-called healthy lifestyles. They often eat outside. They don't eat enough fiber. They may have an intermittent abdominal discomfort, including constipation, bloating. And for this particular patient, she has what is termed as irritable bowel syndrome. The symptoms actually bothered her a lot, and she subsequently did a colonoscopy, which is an endoscopy from the bottom. And the doctor found a growth, obviously a cancer. So, but my question is like, the first is, you know, what is IBS and the connection with gut microbiome? So I would like to start this first in that very often when doctors, at least in the more older days, we tend to have this what I call a separation of mind and the body, where the mind itself is a separate you know, disorder from a physical. So many of our Western thinking process for doctors is that we treat the disorders, but sometimes we don't regard the psychological states. Now for IBS itself, this is a very common disorder, chronic and recurrent, and most importantly, there is an absence of any organic disorders. They tend to have impairment in the quality of life. Bloating is very common, and meal is often a trigger of the symptoms. Now, this IBS is part of what we call as the disorder of gut-brain interaction. Now, many of these disorders are affected by, what it means by the biopsychosocial model, meaning that the disease itself, the IBS, the symptom, are not just because of the disturbance in the gut, but there's often other biological, psychological, and social diseases or social disorders that are often affect the symptoms. And that's the reason why many of our patients who has abdominal discomfort, they are often affected even by stress or emotions, or even if they have a disturbance in their family and also their social life. Let me share a story, a true story in my own home country, where back in 2014, we had the most severe floods that caused 200,000 people homeless and no clean electric water and electricity. So many of them actually fell sick. And this gives us an opportunity to carry out a community studies in 2 of the villages where we collect the stool for metagenomic analysis, and then we administer probiotics to the flood victims. What we found was that out of that 200 flood victims, almost half of them developed new onset of irritable bowel syndrome. And what we found was that those who developed a new onset IBS, they actually have a dysbiosis, mainly these 2 organisms. And these 2 organisms are mostly derived from the environment because of the poor water and sanitation and also hygiene practice. And when we prescribe a probiotics for 3 months, it does help to reduce the symptoms, improving the quality of life. And we believe that this is true restoration of the microbial balance as well as the gut-brain health. So back to the patient itself, again, I need to highlight this. Sorry. All right, so yes, let's go back to this particular patient where, she is obese with a fatty liver. She often eat outside and don't eat enough fiber, and the doctor found a growth. So you may ask a question, what could be the relationship between the colon cancer, the microbiota, as well as the obesity? Now, colon cancer is the 4th commonest cancer worldwide, and is the 3rd commonest cancer among males and 2nd in female. The question is like, why is there a rapid increase in the colon cancer? Now, I would like to change a bit of paradigm for many of us. We know that those who got colon cancer, it's probably because they have eaten less fiber. But actually, in today's world, pollution and plastic, especially plastic pollution in the environment, is a big problem. There are a lot of plastic particles that can be found in our drinking water, even in the seafood and in other food that we don't even know exists. Now, this recent paper that we have published, we have actually found microplastic in human colectomy samples, meaning that these are human colon that have been surgically removed because of a disease like cancers. And it's very interesting. This will be this was actually the first paper where we detect microplastic in the human colon, where many of these because the colon were removed because of colon cancer, and that's why we found more microplastic in them. And it begs the question on, could the microplastic that exists in our diet, in our water and so on, have any effect on the cancer genesis or not? In fact, even from the air pollution, it does actually have an effect on bowel cancer, appendicitis, bowel infection, and even inflammatory bowel disease. And this is because all this air pollution particles may be ingested, and this ingested particles would then lead to a gut dysbiosis, alteration in the immune activation, and so on. So another study to show how potentially pollution may actually affect the gut microbiome. Because of this pollution or particles, it actually result in increase in the alpha diversity, right? Especially from proximal until distals, and also cause an increase in the TNF alpha expression. This is actually an immune modulatory particles that actually increase in response to the air pollutant itself. So what I'm trying to say here is that air pollutions, ingested particles from the air, from microplastic that were found in water and so on, all of this would have a profound effect on the gut microbiome. Also, other than what I mentioned about the diet, and there are also metabolic disorders like obesity that seems to have an effect on GI cancers as well. In fact, many are not aware. Many people would just link obesity with heart disease or diabetes, but they did not know obesity can also lead to cancer. So we have a group of experts again coming together. We discuss, we review literatures and so on, and we did find that obesity is a cause for several GI cancers, including colon, esophagus, stomach, liver, pancreas, and the gallbladder. This is just to illustrate that this is one of the studies that we are currently ongoing, where we want to link obesity to a colon cancer. More importantly, is that we like to find what could be a potential biomarkers that hopefully some biotech company might be interested, looking into fecal microRNAs and how these microRNAs could explain the mechanism that initiate and even progress colorectal cancer in the obese patients. So another slide showing how this traffic air pollution may have effect on gut microbiome in obese individuals. I also have some interest on the artificial sweeteners. For those who are actually taking artificial sweeteners as as a replacement for sugar in diabetes, it has been shown in recent articles that it actually may cause more glucose intolerance by changing the gut microbiome. Okay, let's go back to the patient again. So she asks, what can we do to change the fate? So this will be the last section where I talk about the potential therapeutic applications. So one of which most important therapy would be the use of probiotics. We have several working groups with experts and so on coming up with position statements and consensus. And the reason for doing this is that the literature itself is a bit unclear with regards to the use of probiotics in certain GI disorders. Now, these are some of the statements that come out from the most recent paper that we published. In order for a probiotic to be useful as a drug, the strains should be well characterized and deposited. The manufacturing process for the probiotic should be optimized. It should be properly registered as a drug. The efficacy should be confirmed by a randomized control trials. There must be certain strain specificity that is important rather than combining all or as many strain as possible in the probiotics that is commonly found in many of the probiotics we found in the shelf, right? There are just too many probiotics, but none of them have been proven by any clinical studies. And probiotic is not all safe. There are certainly high-risk population that we probably should not use probiotics, including even especially those who are in the critical illness, in the ICU, or those who have high risk for infection. Now, probiotic can also exist in different forms, in yeast form and bacterial form. Whether or not should we use a single strain or multiple strains, and what are the diseases that we commonly use probiotics? And this include acute infectious diarrhea, antibiotic-associated diarrhea, traveler's diarrhea, and as well as irritable bowel syndrome. So if this patient asks that she wanted to start on a probiotic, but there are just too many choices. I believe this is also the same problem, not just in Malaysia, but also in India as well, where there's just too many choices of probiotics. And very often these are considered supplements. The government don't, they regard this as supplements. So it cannot find or prescribe a probiotic in the hospitals. So how can you actually prescribe a supplement in practice? This could be a question. Also, there are several other considerations. Should a probiotic be cold chain or on the shelf? What it means is that by right, a probiotic should contain live bacteria. Now, if you were to put the probiotic on the shelf, exposed to the sun, the chances are by the time you buy them off the shelf, there are probably no more live bacteria left. And therefore, ideally is that the probiotic should be in a cold chain. And if you do happen to find probiotic on the shelf, how do you guarantee a quality, meaning that, whether or not there are still live bacteria exist? And the thing is that, by the time the patient come to see a doctor, in fact, many of them have tried some form of probiotic or even functional food. And sometimes they find that this product are not really helpful. So how should you advise your patients? So these are some of the common questions that we encounter in practice. So based on those questions, therefore, what we recommend as experts are that probiotics should be registered as drugs. And in fact, any compound that are registered as drugs by the regulation, they mandate certain standards for consistency as well as for quality. So this is something to think about, when for the probiotic industry in the country. Also, as doctors or even consumers, we should choose a probiotic product produced by manufacturers that are able to provide stringent quality control measures. So I think this is very important. And despite that also, we need to consider, the transportations, whether or not, how do the probiotic are being kept on the shelf or in the fridge and so on. So all of these are other considerations that we need to think about. And how do we actually choose a probiotic? Most importantly is when we do prescribe a probiotics, we have to ideally based on the science behind why the choice of using that particular strain, right? And we need to understand the science, the mechanism. We need to have a strong or robust clinical evidence in the form of randomized control trials. And there's also other consideration, including access. Usually cost is the most important reason for not using a probiotic, as well as the other regulatory and issues and so on. So for this patient where he has a Helicobacter pylori gastritis, was given eradication therapy, but he then developed antibiotic-associated diarrhea. So he was started on this particular probiotic, which is a yeast type of probiotics. is the probiotic a right choice? This is just to mention that after we give a eradication therapy for Helicobacter pylori infection, it's very common for them to develop antibiotic-associated diarrhea. And if we were to start on this probiotic, in fact, if we were to sometimes in practice, what we do is like we co-prescribe the antibiotic with a probiotic, and by doing so, it will reduce the effect of if you don't provide a probiotics. So they would, if you were to give a probiotic, more likely they do not have the antibiotic associated diarrhea, and they will also have a much more balanced gut microbiome. Now, for this particular probiotic, it has extensively studied. So this is what the reason why this particular probiotic is preferred, at least in my practice, is because there are many mechanistic studies done that will explain why this probiotic works. Also, they have many randomized control trials that shown how this would benefit the diarrhea diseases. So this is just another table showing what are the others probiotic that may be useful in certain diseases, including acute infectious diarrhea, antibiotic-associated diarrhea, Clostridium difficile-associated diarrhea, as well as traveler's diarrhea. Also, probiotic has been researched in Parkinson disease as well as Alzheimer's disease, given for a period between 4 to 12 weeks. With either single strain or multi strains, and they found that by giving probiotic, it does helps to reduce the gut inflammation and may potentially even improve symptoms. So other than the probiotic, we do have other more invasive method for modulating the gut flora, and this is what we term as the fecal microbiota transplantation, or the FMT. So what we do in FMT is that we ask the donor to provide their feces, and this donor ideally should be the super donor where they are certainly healthy, free from diseases, and when we sequence the microbiome, they are of an ideal profile for someone who is healthy. And after we got the donor feces, we then mix them up, we blend it, and then we filter it. And that filtered liquid, feces liquid, are then injected back into the endoscopy. So this is in the early days is what people do, a fecal microbiota transplantation. And it's very useful for diseases like the Clostridium difficile infection, where in the West, it's commonly caused a severe form of colonic inflammation. As the success has shown, people are now researching into how to use this super donor feces and pack into an oral capsule that can be ingested rather than to spread it during the endoscopy. So I would like to conclude my talk by this slide where the little brain and the big brain is really a very complex bidirectional communication between the central nervous system and the enteric nervous system, which is vital for homeostasis and health. And the gut microbiota is really an important part of this health equation. Dysbiosis can be due to a disruption in the exposome, the diet and antibiotic, the environment, and can cause disorders in the little brain, big brain. The potential therapy to modulate the microbiota gut-brain axis is available, including probiotic. Certainly, we do need further research. So with that, I wish everyone would have a healthy mind, healthy gut. And lastly, we have done some really fantastic research, and all of this is because we have a great, members in my research team. So I'm really thankful and really hope that everyone would also like to collaborate in this research area. Do get in touch with me. again, thank you very much. And that will be all for today.
Thank you very much, Professor Lee. And he explained very nicely about the microbiota and adverse effects about that, the different pollutions and the way of infections. Now the session is open for the speakers for interactive session.
Solution, sir, yesterday నేను ఓపెన్ సార్ నేను ఆల్రెడీ మీ దగ్గరికి వస్తా సార్ నేను.
Now the session is open for the interactive thing, and you can interact with the Professor Lee. Any questions from the participants?
Yes, ma'am. Professor Lee, I would like to have ask you a few questions because I'm gone through your presentation thoroughly. So you said that in the Indian scenario, these Helicobacter infections are very increasing, but stomach cancer is not common. Why? Is there any significant study in this?
Yeah, this is an interesting observation that has been reported by our colleagues from India, where they find that Helicobacter pylori infection is very prevalent in India, but not stomach cancers. Now, we know that Helicobacter pylori itself is associated with a poor hygiene. So there is a reason why certain countries, especially in the Asia Pacific, we do have more Helicobacter pylori, but there's also a genetic component that we are not exactly sure, where it's possible that Helicobacter pylori exists in many forms, the more benign form and the malignant form. Usually the malignant strains are the one that cause cancers. So it's certainly possible that in the Indian population, they may have a less malignant form of the Helicobacter itself, or there may be certain diet or, for example, even yogurt, for example, that may actually protect the stomach of in India from getting the cancers. So this is interesting, but we still need really investigation into that.
Thank you, sir. One more question I would like to ask you, sir. So you said that the infant formula and breastfeeding change the microbiome. So what is the impact of this one in their brain development?
Yeah, very good question. In fact, breastfeeding is probably the best source of food for the infants. It has all the right ingredients, especially the probiotics as well as the prebiotics, right, in the form of what we call as a certain beneficial fibers and proteins or carbohydrates, and certain so-called probiotic, especially like lactobacilli and bifidobacteria, that are very abundant in the breast milk. Now, it's very difficult for infant formula to replicate the breast milk. Many companies have tried to do that, but they couldn't replicate, right? So this shows how unique is the breast milk. And whether or not this is possible, well, we can't say that it's not impossible, that there's so much of research going on. Many companies who are working on infant formula, they are pouring in a lot of money, trying to find what could be the best composition in the infant formula that would have effect on the cognitive development of infants or brain development in infants. Now, one of my most recent interest is the milk fat globule membrane, or what I call as MFGM. Right, MFGM, or the milk fat globule membrane, consists of a lot of these phospholipids like gangliosides, sphingolipids, and so on. And these are clearly beneficial for the brain development. And interestingly is that this MFGM that contains in the breast milk also interacts with the gut microbiome, right? And this is how it may potentially provide benefits to the cognitive development, reduction in infection, and so on. So industry again is trying to incorporate the this so-called MFGM into the infant formula. So whenever they find from something that's benefits from research, the industry, they'll try to rush in, pouring in money to make the best infant formula. This is a really a very interesting race between the various companies and industry out there.
So if you allow me, I would like to ask you 2 more questions. Can I ask?
Yeah, sure, of course.
Yes, sir. Sir, how to overcome this antibiotic-associated dysbiosis? Is there any other preventive therapeutic method other than the probiotics?
Yes, in fact, I did mention antibiotic is one of the I wouldn't say bad boys, but certainly we are over prescribing antibiotics by doctors, by pharmacists, and so on. And the effect of that is that it leads to antibiotic resistance. We need more and more powerful antibiotics. So the companies are all trying to produce their most powerful antibiotics because all their old antibiotic doesn't work anymore. And that is a big problem, right? So rather than using stronger antibiotics, what some people are doing is why not we use probiotics instead, right? Because the antibiotic itself, as I mentioned in my talk, it does disrupt the normal flora or normal microbiome balance, and because of that, it's really not advisable to use or over prescription of antibiotics. And we should instead think about giving a probiotic in certain diseases, for example, diarrhea disease, or we can co-prescribe a probiotic with antibiotic in order to reduce the effect of the antibiotic itself and may potentially reduce resistance. So this is something that even the industry can think about.
And one more thing, sir, you had explained a good point that I have noted that one. The restoration of microbiota is necessary when fruits are affected at locations, but most of the countries are not following the same. I think so. So is it compulsory to restore the microbiota in the flooded area?
Right. Now, many of diseases, some of the GI diseases that we see, like diarrhea, for example, which I also know is one of the main killers in India, but also many other countries in the Asia Pacific, is because the GI disease itself is a result of this dysbiosis or abnormal microbiome that we have, right? So in order to prevent those diarrhea diseases, one is that we need to build a good microbiota profile by providing good nutrition to our children, for example, right? The thing is, like, many of our children, they are undernourished, they are malnourished. So when they are malnourished or undernourished, they don't have the nutrient required for a healthy gut environment. And many of the diarrhea diseases that we see in these malnourished children is because of that reasons. And therefore, it's very important for us to really restore the gut microbiome by giving proper nutrition to our children. And that itself would have helped tremendously, in reducing this disease.
Yes, sir. Thank you, sir. So one more thing, sir, what I observed is you said that the air also affects the gut microbiome. So is there any relation between the respiratory system and gut biome?
Yeah, so a great question. In fact, microbiota doesn't the microbiome doesn't just exist in the gut, but it also exists in other systems, including the lungs, the reproductive system, the skin. So all of these, they have microbiome. Now, similar to a gut, the microbiome in all these systems, they must be in balance, right? Any imbalance may potentially lead to diseases in that system. So same thing with respiratory as well. Like the COVID-19 pandemic that we are experiencing now, right?
Yes.
The COVID-19 does have an effect on the gut microbiome. And because of that, it may actually cause an effect on the respiratory disease or the gut diseases and even neurological disease. So we shouldn't neglect that, that factor of microbiome exists in also other systems. Although the most abundant gut microbiome exists in the gut, but other systems, they may have a different composition and will require, specific research for that particular system in order for us to understand it further.
Okay, sir. So just I would like to have a last question. If you allow me, sorry, I am asking you more questions.
Sure.
Sir, how to confirm that the individual is healthy in the FMT treatment?
Yeah, the definition of healthy is obviously difficult to define, although for in the lay term, obviously it's like they are free from diseases. But this would be tricky because no one can be free for diseases forever, right? I mean, at some point there may be changes to the environment, to the food, to the emotion, to the psychology. All of this may create imbalance in health and therefore causing disease. So the state of health is I think is a very dynamic, very similar to the gut microbiome itself. If our gut is healthy, by making sure a balance in the microbiome through good diet, good nutrition, avoidance of drugs like antibiotics, or use it judiciously, a balanced psychological state, all of this would certainly promote a healthy person, so to speak.
One more thing, sir. Sir, what is the role of stress on the microbiome?
Yes, in fact, the state of mind has a great impact, on the gut microbiome itself. It could be two ways, not just one way. If you have a stress, it can lead to changes in the gut microbiome and therefore cause symptoms like abdominal pain or bloating. On the other way around, if you have a gut dysbiosis to start with, it will also lead to activation of all these hormones in the or even activate the neurotransmitter in the brain and cause stress. Then it will cause a vicious cycle, then the vicious cycle will start, and all this will then leads to even worsening of the condition. So that's why it's very important that I think we need to keep a balance between the two. The gut-brain axis has to be in a state of balance.
So we can say that the microbes are ruling us.
Yeah, exactly.
Thank you. Yes. So thank you very much, sir, for just, I had some few questions and I discussed with you. Dr. Rao is here, sir. Would you like to have any point?
Sir, give chance to others, sir, to like to ask.
Yes, definitely.
Let the participant ask some questions. Maybe later on, I'll take.
Hi,
Hi, sir.
How are you?
No.
Thank you for joining us.
Yeah.
I'm very happy
I was
to
there
see you,
since the beginning, not from the beginning. Maybe I'll just talk after the participants ask the questions.
Yes.
Very happy.
Participants, please.
Yeah, sir, you told about the probiotics are having that effect on COVID infection patients. Is there any related research on that? Probiotics are helpful for recovery of the patients? What are
Yeah, yes, indeed. I mean, there have been, I think, recent research that look into the role of probiotics in the COVID-19. Some of them are really small studies, and some have shown beneficial effect. Some did not, and it all depends on how or what do you want to use the probiotic for. I mean, at the end of the day, right? The probiotics are really good in maintaining health, right? But when it comes to treating a disease, it's a different is a different ball game. It requires a really thorough knowledge of the disease itself and what exactly the changes are to the microbiome. It's not as easy as just prescribe a probiotic, and we hope it prevents the more the disease development or disease progression. But we need to have a more thorough knowledge. And this is something that I think academia is really good at. We are really we ask questions, we try to answer them by doing and conduct studies. And hopefully, once the studies really provide a robust result, and that's how the industry will take it up, which is very similar to what we see with vaccine, isn't it?
Yes, sir. Thank you so much, sir. Now, I invite Reverend Father S. Emmanuel S.J., correspondent of Loyola Academy and FDP course director, for conclusion remarks for this session.
Sorry, sir. Just there is one question from Nishita, I think so, that is posted in the group. Just can you ask the same question for these people? Check one question, ma'am. Nishita, in chat box, if you go to chat box.
Yeah, sir.
Yes,
sir,
you can ask the question. Yeah.
Sir, I would like to ask, what kind of probiotics do we have to use to come out of this IBS, this irritable bowel disease, or even to come out of the post-COVID effects?
Yeah, thank you for the question. Now, for IBS, right, or irritable bowel syndrome, there have been some interest on the use of a bifidobacteria, which is a common beneficial bacteria that we found in the healthy colon. Now, like myself, I mean, the study that I've conducted among the flood victims, I've given a form of a bifidobacterium, and it has been shown to be helpful. There was also another group where they use another strain of a bifidobacteria in IBS, and it has been shown to be helpful as well. Now, bifidobacteria are mainly found in the lower tract, whereas the lactobacillus are found in the more proximal tract. So I think that's one of the reasons why bifidobacteria probiotic really helps irritable bowel syndrome. Now, when regards to COVID-19, I really think that we still need quite a number of research. But if you were to depending on what you are really treating for in COVID-19, if you were to treat, say, for example, diarrhea diseases in COVID-19 with GI symptoms, right? And therefore, even a probiotic like the yeast-based probiotic that I have already shown in my slide may be helpful, but certainly, we do need a clinical trial to support that.
Over to Father.
Okay. So good evening to all of you. I'd like to first of all thank and congratulate our renowned scientist, Dr. Yeong, for his wonderful presentation. It's indeed a rare opportunity. It's a great privilege for more than 168 people who were listening to your insightful talk. Am I audible?
Yes, Father.
Okay. I would not be able to go into the details in summarizing. If I do that, I will be revealing my ignorance. I am not a microbiologist. I am a botanist. And I really, I have noted down few points by which I try to conclude this. Dr. Lee explained to us about the role of molecular technology in understanding the bacteria in the gut. And also he told us that now and then when we take antibiotics, and the antibiotic sometimes causes diarrhea. No, we really don't understand the molecular mechanism and then causal organisms and all that he explained to us very nicely. And with the help of a case study analysis, he was able to throw light on irritable bowel syndrome, how it is really causing problems for us, and also very insightful understanding about the colon cancer. Also, there is a relationship between colon cancer and obesity. And also, interestingly, I just listened to a particular thing like harmful nature of artificial sweetener. People suffering from diabetes know they take to artificial sweetener, which is not really good for us. And important statement which he made, that is, most of the diseases are not merely physical. And the Western idea is to understand human beings as separate compartments. But then the Eastern idea is to look at human beings holistically. Therefore, biopsychosocial concept is really very important for us. That's why we always recommend meditations and yoga and all that, to be at peace, to understand ourselves. And then our unnecessary stress will lead us into a lot of problems. Towards the end of his talk, he explained to us about the importance of probiotics. So with this, I want to once again thank the organizers as well as the presenter for having really made this session a wonderful, fruitful, and meaningful. Thank you, one and all. Thank you very much.
Thank you.
Thank you, Father, for your conclusion remarks. Now, Professor Lee, we like to ask some more questions because participants are very eager to listen from you, their doubts about that.
Yep, sure.
Yeah, the participants, can you ask directly to Professor Lee about your doubts? Kalpana?
Yeah. Good afternoon, sir. It was really wonderful talk. Sir, I'm just curious about the appendix. If you are working on appendix as well, and the microbiota from appendix as well, and its role in the etiology of neurodegenerative diseases.
Yeah, this is a very interesting question. There's has been some interest on the role of appendix. In fact, we thought that appendix is not useful at all. That can be easily removed. That's what the surgeon would think. For those who have appendicitis, they would just remove the appendix. But more recent research has shown that the appendix itself is not useless, but has an abundance of microbiome, and it may even play a modulating role in some of the diseases that we are not aware of, right? So I think there is some research, certainly interest in the research in that area. Although I have not been really looking into this in depth, but I did have come across some of this research with regards to appendix. And you are right, it may even play a role in some of these neurodegenerative diseases, including like Parkinson or Alzheimer's. But how the exact mechanism or the direct, or whether the link is confirmed or not, will is still unclear at this juncture, and will certainly require more studies.
Thank you.
Yeah, thank you, sir.
Thank you.
Karunasri Kosuri.
Unmute yourself.
Karunasri, madam, can you ask? Her question is how to restore gut microbes after COVID treatment.
Right. Obviously, I think a lot of people are interested with COVID-19. Yeah. So, I to be honest, although some of my colleagues, even from Hong Kong and so on, they are researching on the effect of COVID-19 on the gut microbiome, but whether a specific probiotic is more useful is still pretty much under investigation. So I would say that in COVID-19, one important part of therapy is really maintaining health. So maintaining health also means that you should maintain a healthy gut, and certainly as long as you are taking probiotics that will promote gut health, I think it will certainly give some meaningful effect. Although I wouldn't say that what are the specific probiotic that we can use to treat certain effect of COVID-19. That is certainly something that I couldn't able to answer because there are still some ongoing research.
Thank you, sir. Rakesh Gaur.
Welcome.
Sir, you can ask.
Yeah, am I audible, ma'am?
Yeah, now at present it is.
Yeah. Sir, is there any relationship between vegetarianism and non-vegetarianism related to the gut bacteria? I mean, that is the healthy bacteria.
Right. This is a great question. Now, when we talk about vegetarianism, in fact, we are mentioning the use of fiber, right? Because vegetable contains complex carbohydrates, including fibers, and having fibers are actually beneficial for the growth of good bacteria. So those who are taking vegetarians, they do have a probably more Prevotella. As I mentioned in my talk, Prevotella is the fiber degrading bacteria, and it can produce beneficial byproduct like the butyrate, which has an anti-inflammatory effect, right? So taking vegetable is certainly good for gut health, and there are, but at the same time, too much of vegetables may induce bloating because of the content of the carbohydrate itself. Will also allow more fermentation, and therefore some vegetarianism may experience more gas because of this reason.
Thank you very much, Professor. And one more question, Nichita Are. Ma'am, can you speak to Professor? Or I would like to ask question. So her question is like this.
Yeah. I have already asked, ma'am.
Yeah, you asked. Thank you so much.
Now I would like to ask Dr. Rao to say a few words about today's session.
We welcome you, sir. Ishweshwarao, sir. Please come.
Yeah.
Good evening.
Thank you so much, Prof.
Namaste.
So it was really informative session, as usual. I don't need to say much words about it. So I hope the participants have benefited from your talk.
Very much, sir.
Definitely, it is unique presentation where the participants usually don't get this kind of information. And I was really connected when you were mentioning about the yellow floods. So it was actually a terrible time for us. We were in Kotabaru, both of us. So 1 week we were really, we were facing lots of problems. No power, no telephone connections, and everybody was worried. And I am very glad to see the results. From that also you have done some studies, which is actually realistic. And I also request if any participants from medical related field, so you can explore the possibilities for collaborations, and you can take suggestions from senior professors from Prof. B. So that's all I would say. And I'm very much thankful to you for accepting our invitation, to emphasize more on this particular gastrointestinal problems and other things. So thank you so much.
Thank you. Thank you very much.
Now,
Sir,
all the participants.
Sir, one minute, sir. Only just it is a very common question about. I have doubt when we are discussing about medical aspects. So approximately how many microorganisms we can found in our intestine? Not exact, but expectations.
Yeah, this is certainly a question that will not be known the last time because the absence of all the sequencing technology, but with now the sequencing technology that we have, we are able to ascertain that we have not just thousands, but we do have millions and billions of organisms. Now, of course, the numbers that I mentioned here may still be more because sometimes it all depends on the technology that we have. Even the sequencing technology that we currently have, we only allow us to sequence probably 70 to 80%. We still couldn't sequence the other 10 to 20%, right? So we do the technology will keep on developing, and hopefully it will give us more idea on what exactly the whole profile of the gut microbiome. But anyway, there are certain groups that are looking into the profiling of this for different population, and maybe you might want to pay some attention to some of those upcoming publication from those groups.
Thank you. Thank you. Thank
you. Before we end the session, I would like to have a group photo. So can all the participants, I request you to switch on your videos so we can take a group photo.
Thank you, sir.
Yes. 1 second. Yes. Yeah. So once again, sir, from behalf of the organizers, and our sincere thanks to you for accepting our kind invitation. And Dr. Rao, sir, helped us a lot to make things possible today, because most of the faculty members don't have awareness about this gut microbiome, because this is a very interesting field. This interest field at the same time is a very upcoming field, because the probiotics is going to rule the world. As you said that if the FDA approves this probiotics as a drugs, definitely it will give a good impact on the pharma industry. Yeah, I hope so.
Thank you.
Please, ma'am, you can conclude the session. Yeah.
Yes, sir.
Thank you very much. So have a good day, everyone.
Yes, sir.
Sir, vote of thanks. Good evening, everyone.
Yes.
I am Dr. Sirisha. I am here to present the vote of thanks for today's technical session. I would like to thank our speaker of the day, Professor Lee, for enlightening us with his knowledge. The word knowledge is simple for him, and maybe through his wisdom, he gave insights about gut microflora. Today's talk was full of information and interesting facts about gut microbiota and the little brain and the big brain. Sir clearly illustrated about the gut, that is the little brain, lies a complex hierarchy of neural, molecular, and cellular interactions with the central nervous system, that is big brains. A concept of disrupted microbiota, gut-brain axis is especially pertinent in functional gastrointestinal disorders. So inflammatory bowel disorders, but also a number of neurological and physiological disorders. There are like Parkinson's, Alzheimer's, and so on. Sir even gave insights about the air pollution and its adverse effects on microbiota. Thank you once again. I like to, from heart, I'm going to give the thanks to Professor Lee for taking out time from his busy schedule and enlightening us with his knowledge. I would like to thank organizing team who really worked hard to make this event successful. Special thanks to Dr. D. Sambashiva, Assistant Professor of Nizam College, and Mrs. Pushpalata, Government City College. And last but not least, to thank all participants who are present here. I would like to end my speech here. Thank you. God bless you all.
Thank you, sir.
Thank you.
Very much.
Thank you. Thank you.
Thank you.
Thank you, Professor Lee.
Thank you, sir.
Thank you, Prof. Thank you, ma'am. Yeah.
Thank you.
Thank you, sir.